We are getting started with the last session of this exciting Metabolist Day 2026. And our last speaker is Professor Sir Stephen O'Reilly. It's to me a great pleasure and honor to welcome him on behalf of Metabolist Day 2026. Professor Sir Stephen O'Reilly is a professor of clinical biochemistry and medicine at the University of Cambridge and director of the MRC Metabolic Disease Unit in Cambridge. He was trained as an MD in Ireland, Dublin, and undertook postgraduate training as an endocrinologist in London, Oxford, and Boston before being independent group leader in 1991 in Cambridge. There is a lot of contributions to the scientific knowledge in the context of obesity and metabolic diseases that Professor O'Reilly bring to the scientific community, but mostly his role on identification of leptin and leptin resistance in the context of metabolic diseases and endocrine disorders, and how such information can be used in the context of better understand these diseases, treatments, and complications. It's what makes Professor Stephen O'Reilly a big figure in the metabolic diseases. And without further ado, Stephen O'Reilly, welcome to see to
Thank you very much for that very kind introduction, and thanks to everyone, Julian and others, for inviting me back again to this wonderful mecca of metabolism. First of all, I have to apologize for my appearances. I did not get into a fist fight with Matthias Chop this morning, nor did I get drunk last night and fall over. I, in fact, walked through a plate glass window. You are so clean in Denmark, and everything is so that I thought it was complete. Oh, I went to see somebody and walked straight into a plate glass. Anyway, that's hence the plaster. So I had to decide what to talk about today. I got a number of topics in the lab, and I thought I would talk about adipose tissue primarily in the relatively short space of time I've got today. Here's my various conflicts of interest. My scientific life since starting my own lab really has been a tale of three hormones. Most recently, a hormone GDF15, which was discovered in 1997, really only became a hormone when its receptor was identified in 2017. I love that hormone. We're having great fun with that, but I'm not going to talk about it, but I can happily take any questions on what we're doing in GDF15. Leptin, melanocortin system, and the regulation of the central control of appetite and body weight has been a big theme of my life for many years. I'm not going to talk about that either. But the first hormone I ever fell in love with, and I'm an endocrinologist by training and by instinct, and my life has always been viewed through the prisms of hormones. The first hormone I ever worked on was insulin, and I started my lab in 1991 to work on insulin resistance. And that is the topic I'm going to talk to you about today in a bit of a background and a couple of new vignettes that I'll share with you. There have been a few common themes that have run through the approaches that my lab has taken to these questions. And the common themes have generally been like to try and start with Homo sapiens as the major, at least the first port of call, and you always make what we do the experimental organism of choice being Homo sapiens. Not that we don't use mice and not that we don't use other organisms, but we like to see if we can get our information from Homo sapiens. And when we do so, we like information to be causally relevant. In other words, we don't like doing associations because that's really tedious. And therefore, human genetics has been really at the center, because with human genetics, you can start in one direction and move in another, and at least you get a pointer to causality when you use human genetics. And a particular type of human genetics that my lab have found very useful is when you find, even if these variants are relatively rare, when you find truly disruptive variants that have a major effect on phenotype, then you can feel you can get true illumination and some really glimpses into how homeostasis works and how it can be disturbed in disease. So what I'm going to talk about today is really, when we get fat, why do we get sick? Which is, what are the adverse health consequences of chronic overnutrition? When we chronically ingest more calories than we expend, what will happen is we store them as fat, and we store them as an expanded mass of triglycerides in our adipose tissue. Yeah, I don't really like obesity much as a term, but it's in the vernacular, so we have to use it. But basically, all it means is an expanded mass of triglycerides in adipose tissue. We're interested in that because people get sick and people don't do well when that happens to them. And I think it's useful to think about the ways in which they get unwell. And I find it helpful to put it into 3 buckets. And one bucket being the, if you like, the mechanical and gravitational effects of obesity, where the sheer mass or volume of the adipose mass is directly contributing to disease through putting weight on one's lower limbs, through increasing intra-abdominal pressure with reflux, through narrowing the airway. And so there are ways in which the sheer volume and mechanical effects are really doing something bad. But when we come to other effects, like the increase in certain cancers, or the cardiometabolic and endocrine consequences, how does that come from a bag of triglycerides stored in adipose tissue? Or does it? In fact, is it simply the obesity is a biomarker of something else going on, which is the consequences of chronically exceeding one's expenditure by one's intake, and is something else going on to result in the adverse consequences of obesity? So one of the earliest things that happen when people start to become obese and overweight is that they become insulin resistant. Now, that's independent of diabetes. Even if your glucose is completely normal, as you get fatter, you find that you need more insulin to control glucose homeostasis in its 2 key target tissues, the liver and muscle. They are the 2 main tissues in which whole body glucose homeostasis is controlled. So insulin resistance is really important. The concept first brought up in the 1930s by Harold Himsworth, who showed that some people with diabetes need a lot of insulin, some people needed very little, and that was the first concept of insulin resistance. And then in the 80s and 70s and 80s, Jerry Reaven expanded the concept of insulin resistance, saying, hey, we take people who haven't got diabetes and they need a lot of insulin. They tend to have high blood pressure, bad lipids, myocardial infarction, a whole range of bad stuff. And so he defined the insulin resistance syndrome, or the Reaven syndrome, or syndrome X, a constellation of cardiometabolic abnormalities that predisposes to bad things happening. Its relationship between adiposity and insulin resistance is there, very strong, but it's complex. You can see that this is real data from Nick Wareham's Finland study. BMIs on the X, these are non-diabetic individuals, and you can see how with increasing BMI, fasting insulin goes up exponentially. But what you can also see is a huge variance between individuals at what level of BMI they started to get hyperinsulinemic, at what level. So there's a huge variance between individuals. And much of what we've tried to do in the last 30 years or so is explain that variance, why some people can get very fat and remain thin, remain insulin sensitive, why some people get insulin resistant very, very, very early. And I think if you went to a clinical diabetes meeting and you asked the clinical diabetologists why that happens and why do obese people get insulin resistant, they say, well, we've been to the meetings and we've been to the plenaries and we've heard all the scientists, and the scientists have told us what happens. And what happens is big, bad, inflamed fat cells. And it's these meta-inflammation, and that's all these things. And there's loads of wonderful animal experiments that really say that meta-inflammation is the big key driving force. And it's these mystery cytokines that go from the adipose to the liver and the muscle, and they do all the mischief. But I'm afraid that I'm a little bit of I just stand back and say, hold on, where's the human data that supports that? Because if you look at human genetics, and now it's been done in millions of people, there's really, the extent to which you get an inflammatory signature or a host defense signature is very modest compared to the vast majority of chronic diseases like ulcerative colitis, even Alzheimer's, even coronary artery disease. You see a very subtle, if any, inflammatory signature for insulin resistance. And secondly, every pharma company has taken every cytokine and given it in and blocked it in every instance, and they've never had any progress in blocking these cytokine molecules. And there's an older idea, and the kind of older idea is that your big fat cell is not your enemy. Your big fat cell is your friend. Your big fat cell is by far the safest place to keep your excessive adipose tissue because it is a professionally designed cell designed to store nutrients under conditions of excess and then release it back to you when you need it. And the idea is that you only start getting into trouble when you start reaching the limits of your safe adipose storage and you start redirecting nutrients to non-professional tissues. Now, the mechanism whereby that redistribution starts causing it, whether it's ceramides, whether it's DAG, we haven't got time today to go into that, and it's still a very rich and open question. If you want to win your Nobel Prize, crack that one, because I think that is a really still an amazing question still to ask, how those non-professional cells become disturbed by excessive exposure to nutrients. But what I am going to tell you today, that adipose tissue that's rapidly responsive to nutritional state and anatomically adaptable is critical to the maintenance of metabolic health in the face of caloric abundance. And that inherited defects in human adipose tissue provide key examples of the types of molecular process that are necessary and might even be targetable pharmacologically for the improvement of our metabolic status. Now, I started my work on insulin resistance with a different tack and a different approach. And it was the extreme phenotype approach, and it was looking at people who had astronomical insulin for genetic reasons. And I started with Anna, who's not a million miles from here, and Anna was my first PhD student, my first technician, first PhD student, first postdoc, and now she's here at the Karolinska nearby. And Rob Semple later, and David Savage. And between us, we worked on people with severe insulin resistance, and we found a number of novel signal transduction disorders. It was great. It was exciting. We found monogenic disorders. We were thrilled, and that was great. But actually, I don't think we advanced the science very much at all with this. And why didn't we? I think we didn't. Because if you look at people with these signal transduction disorders, they don't mimic anything like all the phenotypes that you get with obesity. Yes, you get insulin, increased diabetes risk. Yes, you get high insulin. But these people who have impaired signaling have none of the other features of the metabolic syndrome. They don't phenocopy what the sort of thing that we're after. So basically, they told us that what you're seeing in common insulin resistance isn't an insulin signal transduction disorder. But was there any situation which did mimic this is what you saw with obesity? Well, yes, because we started, myself particularly and David Savage and Inez Barossa, started studying patients with lipodystrophy, whose insulin resistance was due to a condition where they were not able to make fat cells or not able to make enough fat cells or not able to store fat in fat cells. And those individuals were all severely insulin resistant, no matter what the monogenic cause, and we and others found many of the monogenic causes of this. But whatever the cause, they all had every feature you would see in an obese person who had metabolic disease, without exception. So lipodystrophy was almost a perfect model for obesity, which sounds mad because these people have either no fat or inadequate fat, and yet they're totally mimicking what you get when you have excessive fat. So one of the key experiments, this is an old study, but one I always return to because I think it has been important and influential and pretty canonical. David Savage and I sat down one day and said, let's talk to our population genetics colleagues and let's chat to them and say, what does it look like just to have syndrome X type biochemistry in the general population? High insulin, but corrected for BMI, high triglycerides and low HDL, all as a cluster. And what are the SNPs in the genome that associate with that? And this work led by Luca and Rob Scott at the particular, but many people involved in this study. We did that, and this. At the time, this was a very large study. Now people Ruth will laugh at the sizes of these studies in those days, but it was one of the first large-scale studies of this type. And what we really asked was, of the SNPs, of the variants in the genome, which tissues are they most expressed with? If you did, as Ruth did earlier, if you did a study on obesity, you suddenly find they're brain genes. But if you do a study on this metabolic syndrome, you find almost no brain genes at all. You find almost everything is in the adipocyte and adipose. All the genes are influencing function in the adipocyte and the adipose tissue. And if you manipulate those genes, you do influence the amount of fat stored. And then the other remarkable thing was, if you yes, it's in the fat cell, they must be the genes that give you the big fat belly. And they're the genes that give you the visceral increase in visceral adiposity. So let's go and take those SNP scores and let's take them into people who have had DEXA scans in their legs and let's ask, do they have big fat bellies? And the answer was, not really. They have a little bit of increase in visceral fat. What they really have is no fat on the buttocks and no fat on the legs. Those risk factors are reducing the amount of fat. It's a bit like Sherlock Holmes' dog that didn't bark in the night. The key is the clue that it's giving you is in the absence, not the presence. So these risk factor genes were diminishing the ability of people to store fat safely in those safe depots in a little bit like a formed frust of lipodystrophy. So we've seen all these monogenic lipodystrophies, and now we were seeing in the general population a similar mechanism. People unable to put those fat in the lower body. So the genetic variants associated with hyperinsulinemia, dyslipidemia, highly enriched in adipocyte expressed genes, and the risk alleles strongly associated with reduced subcutaneous fat, especially on hips and on thighs. Now that's got very general, and now I want to get more specific because we like to get deep into molecular understandings if we can. So I'm going to give you just a one example, and this is a lovely example. This was actually led by my colleague David Savage in collaboration with Corinne Vigouroux, but I think it's one of the nicer examples of how a rare monogenic disorder can give you a little crystalline insight into something that's very important. This is a rare syndrome. It's a rare autosomal dominant syndrome. It's due to a particular type of perilipin 1 mutation, and it has to be a particular type. It has to disrupt the C terminus, put in an aberrant extension, and actually be expressed in adipose tissue as an aberrant protein. And there's been about 12 families now with that, and they all have severe. And remember, these girls have all the features of metabolic syndrome. They have fatty liver disease, they get liver cancers, they get heart disease, they get everything you would expect associated with obesity-associated metabolic disease. Why do they do that? What is perilipin 1? Well, the beauty of perilipin 1, again, in terms of getting cleanliness of biology, perilipin 1 is only expressed in white adipocytes, not even brown adipocytes. It's a white adipocyte only gene. Great. So that's getting us somewhere. What does it do in white adipocytes? It sits on the surface of the adipocyte triglyceride droplet, and when you're being fed, it does nothing, because it just sits there with its partner, CGI58, while your lipid synthesis and triglyceride synthesis puts lipid in. Where it starts to act is when you go to bed at night, when you haven't eaten for a few hours, and when you get your growth hormone spike, and when your heart, which is beating overnight, needs free fatty acids being delivered to it from your adipose tissue, which is helpfully delivering the free fatty acids that you need for cardiac metabolism overnight. And there's beautifully coordinated synthetic breakdown pathways coordinated by phosphorylation events, so that you see the phosphorylation event here at the end of perilipin 1 kicks off CGI58, which then grabs the initiator lipase to cause free fatty acids to be broken down. And then you have HSL coming to the N-terminal phosphorylation site. So a beautiful coordination of triglyceride breakdown to go. Now, why are these people sick? Why they're sick is that throughout the day, irrespective of feeding or fasting, they have a continual imbalance between, they have no ability of CGI58. The amount of it is excessive for the amount of functional perilipin 1. And you're perpetually, in effect, infusing free fatty acids into the individuals throughout their 24-hour period. And that causes every feature of the metabolic syndrome in every individual. So lipolysis and the control of lipolysis seems really important in adipose tissue. And, the control of lipolysis is exquisitely controlled by multiple inputs, both stimulatory and inhibitory in the adipocyte. So you won't be surprised that when we get to the next era of the human genome studies done by many groups around the world, including some from Claudia and others that we collaborated with, but many people have done this. When you start doing rare variant analysis and start asking things, if you use waist-hip ratio as the surrogate for abnormal adipose distribution, you start seeing a whole range of signals for things relevant to lipolysis. ALK7, the anti-lipolytic receptor, PDE3B, which degrades cyclic AMP and regulates lipolysis, PLIN1, which I've just mentioned, amblin for insulin receptor, a potent anti-lipolytic agent. So many of the signals are focusing down on lipolysis. So I'm just going to tell you a new story about an anti-lipolytic signal which we'd come across recently, which has kind of been hiding in plain sight due to a genetic accident. But I'll tell you the story whereby we've uncovered that recently in a collaborative work with Kendra Benz and others at Pfizer that we've been doing for the last couple of years. And this is around the group of receptors, the hydroxycarboxylic acid receptors, two of which, 1 and 2, are anti-lipolytic. HCAR3 is only in higher primates and it seems to bind octanoate. So you may, of course, you're very familiar with its ligand beta hydroxybutyrate, a well-known ketone body. What people are a little bit less familiar with is that ketone bodies are known to regulate their own production, and it makes a lot of sense that they do. As you starve and ketones bodies goes up, you don't want that to happen indefinitely because you get ketotic. So one of the feedback is that you get beta hydroxybutyrate binds the HCAR receptors on adipocytes and suppresses lipolysis. So there is a known feedback loop, which is probably not enough in the textbooks, but is actually a known piece of physiology, that the anti-lipolytic actions of ketone bodies are part of that homeostasis of maintaining ketone bodies within safe limits. Now, what took our attention was that all of these three receptors are in a small area of chromosome 12. And there's a pretty strong signal for things like waist-hip ratio, triglycerides, HDL, all clustered in that region of chromosome 12. And in discussions we had with Hayin Klimt at Pfizer and Kendra Bentz and JP Fortin, we kind of figured we got to figure out what's driving this signal. Because if you go to UK Biobank and simply ask, or you go onto GeneBAS or you go onto AstraZeneca portal, it tells you, we know the signal. The signal is coming from a variant in HCAR3. It's coming from this missense variant, this RC311. Very strong. So this is the lead signal. It was a bit odd because there was another RC311 that was attributed to HCAR2. And if you looked at different databases, these were differentially attributed to both genes in different places. And when you started looking at things like Hardy-Weinberg equilibrium, they were grotesquely out of Hardy-Weinberg equilibrium. There was no homozygotes for any of these, looking weird. And that was puzzling. And the reason for that is that HCAR2 and HCAR3 basically evolved yesterday in the human genome. It's incredibly closely related to HCAR. It's only in primates, and it's really very, very, very closely sequence related to HCAR2. So we in our lab, Brian in particular, Cara Rainbow, and I will mention that tragically at this point in his first year of his PhD, Xiaoming Liu was very heavily involved and tragically died in an accident in his first year of his PhD. So I was dedicate our talk to the memory of Xiaoming. We developed specific genotyping assays for R311C to discern whether it was in HCAR2 or was in HCAR3. And so what we did was we actually developed a genotyping assay that was unique to 2 and 3. And what we could find is that it was in the ketone body receptor, not the HCAR1, not the HCAR3. So it was 17% of Europid genomes effectively were heterozygous for this, and nobody carried it in HCAR3. We couldn't find anybody. So if you go then to the UK Biobank, what the sequences told you it was, but then you corrected it for saying that was just a mistake and everything's in HCAR2, it made perfect sense. It lined up. UK Biobank now lines up with what we directly genotype. So that gave us the confidence to go back to UK Biobank and say, okay, this is what it is now. It's actually HCAR2. What's the data look like in population terms? And I'm going to go through this quite quickly. And I know for population genetics people, these sorts of things are very familiar. For those of you who are not, it's a bit unfamiliar. Basically, the greener it is, the stronger the positive signal. The purpler it is, the stronger the negative signal. And this is this is adjusted for BMI, unadjusted for BMI. You can see there is a BMI signal, but there's a stronger waist-hip ratio adjusted BMI signal. But basically, carriers of this have got a higher waist-hip ratio, they've got higher triglycerides, they've got increased liver function tests, they have a slightly raised glucose, but bizarrely a lower hemoglobin A1C, and that is actually explained by a red blood cell phenotype. It's not a glycemic phenotype in this. There's a strong signal for reticulocyte count in the area. And then if we go to individual populations and look in more detail we'll find that the carriers have reduced gynoid fat, reduced leg fat, markedly reduced adiponectin, and the trend towards higher NEFAs. But when we get a dynamic test of NEFAs, we see that individuals have got an unsuppressed NEFA significantly in the Fenland study, where there's about 800. It's not as a large a study as we'd like, but we see that NEFA levels do not suppress normally after a meal in the Fenland study. And with assistance of our wonderful molecular modeler, Vladimir Saudek, Vladimir predicted that this variant would change helix 8 in HCAR2 and render it being less in less stable membrane apposition. He predicted that this would be not as tightly opposed to the membrane. This change would cause an alteration in membrane stability. And that's indeed what our colleagues at Pfizer, JP Fortin and others found, that the variant has less stable cell surface expression, even though the mRNA levels are similar, and it is more rapidly endocytosed. So this just is an endocytosis assay. The mutation causes the gene to internalize more quickly, causes the protein to in. And the animals are insulin resistant. So if you knock in the homozygous knock-in, animals are insulin resistant on ITT. And they post-meal, like the humans, they don't suppress the NEFA levels as well as the humans do. And if you take the fat out from the animal and you do niacin binding, niacin binds to the HCAR. It's an artificial ligand for the HCAR2 receptor. You can see the reduced binding consistent with the in vitro data. And you can see that in response to beta hydroxybutyrate, you have lower amounts of suppression of lipolysis, of NEFA levels that in response to beta hydroxybutyrate in the explanted fat. So, in conclusion, there's a common missense variant present in 17% of Europeans. We see it in about 6%, 6 to 7% of Asians. It's less common in African populations, but it associates with multiple features of the metabolic syndrome, most likely attributable to impaired suppression of adipocyte lipolysis by circulating ketone bodies. So your ketone body signaling in adipocytes seems to be necessary for metabolic health and not simply an emergency signal in states of starvation. Even relatively simple perturbations of ketone body signaling in adipocytes is enough to predispose you. I mean, the effect sizes here are small. These are modest sizes of effect, but because it's so common, you can see the signal quite markedly. And so is a common variant in adipocyte GPCR impairing controlled lipolysis is a significant contributor to the human metabolic syndrome. And so I'm going to move on to the last bit of my talk and the second sort of new story that I will tell you. And this one always amuses me because when we wrote our last grant to welcome, I told my colleagues we'll have to change tack. I think we've plucked all the low-hanging fruit. We're not going to be able to find any new monogenic diseases. We have to do something different. And then Yanislava Karasheva from Michael Roden's lab came and I said, did you see any interesting patients last year? And she said, yeah, we had these two siblings whose parents were highly related, Turkish of origin, and they had lipodystrophy, and we couldn't figure out what was wrong with them. I said, sounds interesting. And they said, did you do all the exome sequencing? Yeah, we did everything. We did all the genes, and did no cause. I said, that sounds interesting. So these are partially lipodystrophic sisters. They have high insulin, high triglyceride, low subcutaneous fat, high liver fat. They have plenty of visceral fat. So it's not a total lipodystrophy, but very marked phenotypic and metabolic abnormalities and a partial lipodystrophy, mainly affecting the subcutaneous tissues. And so we got ethical permission to get hold of the data, and we interrogated the existing exome data. Simply, first of all, asking Brian Lamb's, a brilliant colleague in my lab, Brian, just have a look at the homozygous stops and see if anything interesting smells in the homozygous stops. So we sat down and we smelt and we sniffed at 5 of the homozygous stops, and 4 of them didn't smell interesting. They were either testis-specific genes or they looked a bit like pseudogenes, but one of them had a smell, and that was LAMA4. And LAMA4 was interesting because I'd never heard of it first one, but it was interesting because at least it had the good grace of being highly expressed in adipose tissue. And actually, locally, Carl Tryggvason, not too far from here, many years ago, knocked it out and didn't say, I've got a lipodystrophic mouse, but he did report saying, oh, look, I've got a mouse that has a decreased capacity for adipose tissue expansion. And so he showed the knockout mouse years ago that here's the normal epididymal fat pad, here's the epididymal fat pad from a LAMA4 knockout mouse. Now, he didn't report any metabolic defects in these animals, and sadly, when we did our own knockout, we couldn't either. So there's often a human-mouse discrepancy in some of these. So we didn't actually we got the same phenotype, but we couldn't make them very insulin resistant. They still had. So anyway, what is LAMA4? It's a key isoform of a major basement membrane protein. I mean, LAMA4 is about the second most abundant protein after the collagen in basement membrane. So it's a very abundant basement membrane protein. And it comes in, every laminin comes as an alpha, beta, and gamma trimer. And LAMA4 is very selectively expressed in adipose tissue. You can see how high this is thanks to Michael's nice adipose tissue atlas, which is all I find. Thank you to Karolinska for providing this lovely data set that we all benefit from. And you can see that very highly expressed. And I assumed that all extracellular matrix stuff going on in adipose tissue would be made in fibroblasts. I thought the adipocyte would do nothing. This is all made in the fat cell itself. This particular isoform is adipocyte generation. It's generated as the fat cell makes itself, and it comes on both at the gene and the protein level in the adipocyte. And in animals who are obese and in humans who are obese, it's upregulated. Its gene is upregulated. So what's wrong with these sisters? They have a total truncation at the very beginning of the gene, so they should really have no LAMA4. And when we look at their adipose tissue and we measure it by Western blotting, there's no protein. And when we do it by immunocytochemistry, there's no protein. So they really are true LAMA4 nulls. But we were a bit stuck. One family, 2 siblings, and a knockout mouse that wouldn't behave itself and wouldn't make itself sick. And so we didn't really know quite where to go. But fortunately, we got in touch. Well, we were managing to liaise with people outside, and we found with Larissa Atahide and Renan Montenegro, we found 2 independent families in South America, in Brazil, who different, unrelated, different mutations, but all stop codons, homozygous frame shifts, either one or the other. And we found 3 individuals from 2 families with the almost identical phenotype and knockouts of LAMA4 due to different stop mutations in LAMA4. When we knock down LAMA4 in 3T3L1s, and we're currently doing in human adipocytes, it doesn't change gene expression for things like adiponectin and CEPB alpha. It doesn't alter that, but it does make fewer cells, and those cells accumulate less lipid droplets. So it makes the cells unhealthier and less like adipocytes, even though their gene expression pattern is unchanged. And as I say, we have human data just coming on this. Very much involved in all of this was a brilliant young research fellow in my lab, Sam Lockhart, who's now back in running his own small lab in Belfast. And Sam had the idea that to take this further than LAMA4. And he did so with, I think, an inspired idea. He said, why don't we look at extracellular matrix in more detail? And then why don't we just think more broadly? And what I'm going to do is I'm going to go to UK Biobank and all of us and take the biggest data sets. I'm going to go and divide extracellular matrix proteins into those that are enriched in adipose tissue and those that are not. And then I'm going to look and see if there's an excess of mutations in when it looks at weight, when you're looking at waist tip ratio, do you find any changes in waist tip ratio when you have an excess of mutations in ECM genes? And so reassuringly, when he did it with genes that weren't exposed to adipose tissue, he didn't find anything. But when he turned to genes that were expressed in adipose tissue, he found 5 significant hits. And I haven't got time to show you all the data, but these are not only associated with waist tip ratio, but all the metabolic features go in the same direction. Even though they're rare variants, the metabolic features go in the direction that you would expect. And indeed, we have now found a single individual in the Genes and Health database who is homozygous for a COL5A3 knockout, and they developed diabetes and hyperlipidemia at age 20 with a BMI of 20. So in the data sets we have others, and COL3A1 is the vascular Ehlers-Danlos syndrome gene, and Aggie Jakubowska in my lab, a clinical biochemist, has now had several such patients into our clinical research facility, and essentially these people have undiagnosed partial lipodystrophy. They already have trouble from their aneurysms, but it's almost universal that they have previously unrecognized partial lipodystrophy. So we're finding collagen genes, we're finding genes for matrix metalloproteinase and other genes in adipose tissue, in the adipose tissue matrix, rare variants of which are really profoundly affecting waist tip ratio. And beyond that, if you start looking at common SNPs in the genome and ask and classify by ontology what type of genes are they for common variants, extracellular matrix structural constituents come out as the top enriched hit for waist tip ratio in the entire genome. Extracellular matrix organization for the third, etc. So if you then do a global look, nearly 12% of ECM genes are linked to a waist tip ratio BMI locus, and that's 10 times more than any other randomly selected gene in the human genome. So I think I'm happy to say, I think we've got a new class of human metabolic disease, which I'm calling adipose matrixopathy, Rare loss of function mutations in adipose basement membrane printing cause a rare recessive syndrome. Rare and common genetic variants affecting ECM contribute very significantly to fat distribution in the general population. Adipose mass and volume are remarkably plastic, of course. You just step back and think, of course, this is bound to be true, because there's no other tissue that we have that has to expand by 2 orders of magnitude, or can do, over our lifetime in an individual. We don't have any tissue as plastic as our adipose tissue. And so it also has to be very plastic, facing during our evolution times of great privation and times of great excess, and be able to protect these lipid-laden, fragile adipocytes while doing so. So it's kind of not surprising that it's susceptible to an intrinsic deficiency in any of its extracellular matrix components. So you'll be familiar with the condition osteogenesis imperfecta, which is basically a disease of bone due to a primary defect in bone matrix. And with slight tongue-in-cheek, we might be able to call this adipogenesis imperfecta, a primary disorder of adipose tissue biology coming from the extracellular matrix, not intrinsic necessarily to the adipocyte itself. So in conclusion, metabolic syndrome occurs when the capacity of adipose tissue to safely store and release energy and free fatty acids in a manner highly responsive to organismal need is exceeded. The greater the extent of the chronic positive, the more we have positive energy, the greater the demand on adipose tissue. That's why GLP-1s work even in lipodystrophy, because if you can take off the load of these embattled adipocyte storage, you still get some improvements. Inherited variation in genes controlling adipose tissue function is an important determinant of susceptibility and resistance to adverse metabolic consequences of overnutrition. A substantial number of the variants impact on lipolysis, that's a really attractive pharmacotherapeutic target, and surprisingly large contribution made by genetic variation in the adipose extracellular matrix. So I'd like to finish by thanking all my colleagues. I mentioned them all through and through through the talk. I thank our funders, but particularly our referring physicians and the patients and participants over the years, without whom we would not have had a research program. So thank you for your attention.
Thank you very much, Steve, for an amazing presentation. Now the session is open for questions. Over here.
Talk.
Steve, why are the excess release of free fatty acids from the adipose tissue harmful? And what's the mechanism behind that?
That's what I said. You weren't listening. You'd win the Nobel Prize when you found that out. I don't know is the answer.
I thought you had it.
That's what I told you. That's the one bit I have not had a chance to talk about. I mean, there are numerous hypotheses. There's the theramite hypothesis. There is the DAG hypothesis of Jerry Schulman. I am not sure. The trouble is, it happens very quickly. It's a very evanescent phenomenon, so it's hard to track down. I mean, Gunter Boden did that 6,000 calorie one day overfeeding study a few years ago. He made people insulin resistant in 24 hours, way before you saw any changes in inflammation or So the understanding. So I think I understand up to there, but now that's for my next generation of people to figure out that's what I'm telling all these young people here to go and figure out for the next. However long it takes, because understanding that a nutritional exposure to, it may be that we've forgotten more than we've learned, because right back in my old days, Nick Hales and Philip Rand, the Randall hypothesis of substrate competition, we kind of dismissed that over the years because Jerry showed that it was glucose transport, not glucose oxidation, that was bad. But I think we may need to revisit some of the concepts of substrate competition. I think there's some elements of substrate competition that are still probably real and going on.
Thank you.
Yeah.
Hi, just over here, up here. To your left.
Sorry, over there first. I'll do that first before you, Clive.
That was a fascinating talk. So I'm actually curious, especially this topic that you presented, where there's a moment where these very specialized fat cells that are doing what they should do, they can no longer store that much fat, and then it kind of bleeds over to other tissues. Do you think there's some sort of range that maybe it's genetic predisposition to how much, or what is this actual range where you're in the red numbers, or it's dangerous?
I think we probably are born, or at least and potentially prenatally acquire a certain fixed ability to create these safe storage, and we sometimes just exhaust. We we don't have an infinite capacity to expand adipose tissue safely, and what influences that is likely genetics and early environment nutrition. Why people from South Asia, often South India particularly, get metabolic syndrome at BMIs of 23 and 24 when there's very little adipose. Is it because they simply cannot generate any new adipose safe storage? So that again is another fascinating topic for further.
And would evolution help us understand these mechanisms
Yeah, it's
in other organisms, perhaps?
because mostly during our evolutionary past, we didn't go through too many cycles of huge nutritional overabundance. So we have got a more unusual environment in the last 100 years or so that we hadn't had to cope with before.
Klaus von
Yeah.
Geneva and Malmö, thank you for a lovely talk. Fantastic. It's always a lean back and enjoy the flight when man hears.
I hope it's not a completely a crazy flight, but
It's fantastic. But I have a comment to the question before on free fatty acids.
yeah.
The Orion Shirihai, among others, has shown that free fatty acids, which should be high in starvation and low when between starvation, are actually high, and they induce insulin secretion, even at low glucose concentrations.
Yeah.
And that is one way into hyperinsulinism, which could be the way into insulin resistance. So I think to lower free fatty acids would already be protecting the beta cell from secreting too much.
insulin. Yeah. Okay. Thank you.
That's just a comment
Yeah. Well, thank you. It's a very
from the islet
yes,
islet
of course.
perspective.
And yeah, absolutely. And the idea that I'm not. I have in the past argued against the idea that hyperinsulinemia can be primary, because I suppose as a clinical endocrinologist, if I see someone with primary hyperinsulinemia, I know them because they're hypoglycemic and fitting, and so it's hard to conceive how you can have primary hyperinsulinemia in the face of going through a phase where you start with hyperinsulinemia. That to me means you have to be hypoglycemic, because how can hyperinsulinemia beget insulin resistance without going through a phase of and of course as human beings we oscillate between we're so close to the border of neurodyspepsia. Normal glucose is 3.3, and at 2.9 we're a bit crazy. So we're so close to neurodyspepsia all the time that I worry about primary hyperinsulinemia as something that we would tolerate evolutionarily. But
But if at the same time you secrete glucagon, which often happens, then you wouldn't have hypo.
it's true if you could manage at the same time. That is true. Yeah.
And that happens in the stimulated islet problem.
Okay. Good point.
There's another question on your first a second. I'm coming, Sophia.
Whoa. Out in the far, the back would be on.
Hi, Sophia from CBMR. Really wonderful talk. I absolutely love the aspect of the ECM involvement.
Good.
I have a question though.
I'll ask you to be the reviewer. I'll ask you, what's your name? I'll ask you.
I'm just wondering if you say like there is an enrichment in adipose tissue, did you have the chance to also look at like certain cell populations that this would link to? If you did not, what would be your hypothesis about this?
So what we do see is that the ones where we see the big signal for metabolic abnormality are much more enriched in subcutaneous than in visceral adipose tissue. So that we do know that most of them are like sometimes 10 to 20-fold higher in subcutaneous. So there's something very special about subcutaneous adipose tissue. That not all of them, but most of them are strongly different in subcutaneous compared to visceral adipose tissue. We haven't looked in any more depth. This is all pretty recent, so we're still.
Okay.
And I'm on a very strong steep learning curve for extra. I knew. If you told me last year, I would say ECM is about the most boring bit of human biology, and now I have to find myself excited and
I'm glad you changed your mind.
yeah.
Did you also have the chance to look at some single cell data? I'm just wondering about also not only the depots but also like the actual.
The individual cells.
Yeah.
I can't tell you that yet. I mean, I'm not. I can't. I no. Great question. Need to do that. Yeah. Don't know yet.
Okay, thank you.
We have time for 2 short questions, one here and one there.
I'm coming.
Yeah, it's coming.
Whereabouts? Sorry. Ah.
Thank you. Great talk. Yeah, my question is, can we just simply speculate that overexpression of this ECM target can prevent insulin resistance by increasing plasticity? Or
That would be great, wouldn't it? Be a brilliant drug target.
exactly. Where is the therapeutic approach here?
Make your nice yeah. But
Is it just working when you have the default, or like in the normal people with no variation,
Yeah.
no variant, will it work?
it would be fascinating. I mean, what we're planning to do and want to do is do a variety of manipulations unilaterally in mouse adipose tissue, manipulating these in overfed and underfed to see whether a limiting amount can prevent or allow further expansion. That's all stuff we're planning to do for the next few years if I'm still alive.
Great, thank you.
Over here. Hi, I'm Olga Goransson from Lund University. Thanks for a fantastic talk. I was interested in the molecular mechanisms underlying the effects that you had when you knocked out the laminin 4 in the isolated cells. So I was just thinking that is there a basal lamina in an in vitro cell system like that? And I mean,
So we've
I'm
done
just thinking that it is the role of the laminin in the basal lamina that would give the effect. So does it make sense to knock it out in a system where you might not have the other basal lamina components?
So we've looked into the adipose tissue, which is diminished from the knockout mice, and so that gives us an in vivo context. And we've got some pretty experienced electron microscopists. And at one stage, we kind of convinced ourselves that the lamina lucida was a little bit, but we couldn't really convince ourselves that we could see any. We couldn't quantitate a difference in the physical appearance of the basal. I mean, what I wasn't aware of before is not. And you may think I'm very stupid. I thought all cells had basement membranes, but no, adipocytes actually do, but many cells don't. And so the adipocytes have a basement membrane, and they still do in a visible basement membrane in the lamina 4 knockout animals. So it's not obviously causing total dissolution of the basement membrane. I mean, you will probably know that the basement membrane is not only important for just structural integrity; it carries a lot of signals between the ECM and so the numerous possibilities is that all of these different ECM genes are having their effect on fat cell health by different mechanisms, by either a physical protective or a signal capacitation, integrins, etc. So we're just at the foothills of trying to understand.
Is it all secreted? Could it have a role inside the cells?
That's possible. As I said, there's no obvious change in any of the gene expression for adipogenesis. So it looks like all those processes are going on. And we will have better. We're doing work in human 3D adipocyte culture. All of that. And we're doing all that right now.
Okay.
And we should have better. 333L1 is a poor substitute for doing it but that's what we just have. That's what we happen to have right now.
Okay. Thanks a lot.
All right. Thanks a lot for the lively discussion. Thanks again for the presentation. Please give me to congratulate.
Thank you.
Have a lovely
Thank you.